So we talked quite a bit about natural selection, but what we're going to try to do today is talk a lot more in detail, and I think what I think is one of the coolest examples of a demonstration of natural selection that involves sort of includes all of the key characteristics: variability within populations, variability among populations, sort of a very good understanding of the genetic basis that leads to the variation in the phenotypes that are under selection, and a clear demonstration of how predators select for these coloration patterns. So here are the goals. I'm not going to spend how natural selection causes what we call microevolution within populations. And what we mean, microevolution is real simple, small evolution. So we're looking at how evolutionary changes occur on a small time frame within a population. That's what we call microevolution. Macroevolution is sort of fossil record, large time scale evolution. Here we're primarily just looking at these sorts of things. And what we're going to really focus on is how particular environments favor particular phenotypes, okay? And that's what we're talking about. How does selection act on particular phenotypes, because it increases a phenotype's survivorship and or its reproductive rate. And that's what we'll be talking a lot about that. We're also going to talk about there are really constraints or limits on natural selection to create, I shouldn't have said perfection, okay, but to create perfection. But oftentimes traits have a benefit in some respect. They often have some negative effects on other traits, and we'll talk about some of those things. And then I don't think we'll ever get to this in this lecture, but maybe I'll plow through it. How phylogenetic trees can be used to examine the evolution of complex traits. And if I don't get to it, I may just have you read or look at it, or I'll do a real quick video recording of how the origin of feathers in modern birds probably originally arose for another reason, okay, in dinosaur ancestors. So we'll talk a little bit about that. So those are the main goals. So one of the things we can demonstrate natural selection really easily, and there are thousands of examples of it. And I focus on a lot of things in terms of extreme environment. We can really look at how extreme environmental effects can cause phenotypic evolution via natural selection. And what we're going to be looking at is, and again, I should have read the textbook ahead of time, but what is California, was experiencing for quite a while in the past 6 to 7 years? An extreme drought, right? This is usually associated with these climatic patterns, and I think it's El Nino, or it could be La Nina. I think it's El Nino. Does it say El Nino? Okay, good. I should read. So this was not the most recent extreme environmental event. This occurred from 2000 to 2004. California experienced also a drought period due to El Nino, okay? So these are Mediterranean climates, and this is a common phenomenon of having these drought periods associated with El Nino. I think what most climate scientists would say right now, in fact all of them would say, is that in fact these are natural events. It's the question of how extreme they're becoming, and are they becoming more pronounced in terms of the degree of the drought, the length of the drought, and all those sorts of things. And I think that's what a lot of climate scientists are trying to understand. But let's go back and look what happened. When you go to California, what they did, this was a great study. These are wild mustard. It's called brassica. You can see they're pretty common, aren't they? So they seem to be fairly common. But during this period from 2000 to 2004, there was an extreme drought in the areas where they lived. The way the pattern worked, there's a lot of rainfall during the winter, often early spring during normal years, and then, in Mediterranean climates, in California, there's not much rain from March until November, okay? So they have drought periods anyway, right, during this Mediterranean climate. But what happened during these extreme droughts is that the availability of water became really reduced during the growing season, right? So they germinate, they grow up, and by the time they got to the normal period, they were running out of water. So there was a really extreme absence of water. And so the question is, what happened to this descendant population that went through these four years of extreme drought? And the trait they looked at was the days to first flowering, okay? So how long, on average, did it take for this population that went through this extreme drought to first flower? And then they have flower, pollination, seeds, and all these sorts of things, okay? So what happened in this descendant population, this was after 4 years of the drought. This is the mode or median, I can't remember what they used, but the average day to first flowering was about probably 46 days, okay? One of the great things is these research had access to seeds from, they'd been working in this population for 20 years. They actually had seeds from the population, from the time period before the drought started, okay? And I'm not going to go into all the details of how they did the experiment, but they were actually able to take all the seeds from that descendant population that went through 4 years of drought, and this was the pre-drought ancestral condition, and they were actually able to germinate those seeds, mate them. They could produce these hybrids between these two populations. They had pure matings between the ancestral population and then also within the descendant population. So what happened here? As you look at this graph, what happened? What was the response? They flowered earlier, right? And that's what, most of what I'm going to tell you is, I always said, you know, what we do in biology is tell you what's obvious, right? Okay, but it makes sense, right? So what happened? Those individuals that flowered earlier, why do you think that genotype or those genes that contributed to early flowering, why were they favored? Well, they survived better, right? They produced more seeds, right? Those individuals that waited maybe on average 60 days or maybe, you know, the water became less available, they couldn't produce seeds, they died, all those sorts of things. So it turns out this earlier flowering was really favored, okay? So selection led to much earlier flowering in the descendant population because those kinds of individuals had a much higher reproductive success. They probably produced more flowers, produced more seeds, all those sorts of things, okay? So that's the mechanism by which that shift occurred. And that, you know, doesn't seem like a whole lot, but you're basically talking, it shifted pretty quickly in about 4 years from about 60 days on average to around 45. So much, that's what we call it, that's what we call a response to selection. That trait changed in that population in response to that, okay? So, and we imagine climate change will probably lead to a lot more of this. And I think one of the things we always think about in climate change, right, is that, you know, obviously organisms can adapt and change, and I think that's an important thing. But I think climate scientists, evolutionary biologists, ecologists are concerned about extreme events, and certain organisms might be able to respond, others might have a more difficult time responding. And when you think about this, this is an organism that obviously it's extremely plentiful, isn't it? It's all over the place. What about a plant or an animal that has a very restricted range, doesn't live, you know, it might be more prone to not being able to adapt to those sorts of changes in climate. So, okay. Does that make any questions about that? And your book has much greater detail about how they did the experiment. This was all an experimental study, and created some really good results. Okay, let's talk again. We've kind of gone over this a little bit, but what we're going to, as we talk about natural selection, what we're going to say is pretty simple. And keep, as I put these things, selection acts on individuals, right? Certain individuals survive, some reproduce, some not. But what we're really looking at in most of the cases is, but selection acts on individuals, but what we're really looking at is population level phenomenon, but causes populations to change in a particular trait. Like in the previous example, early flowering time decreased quite a bit. So that's the trait we were looking at that was under natural selection. And we asked what happened in that particular population. We could look at other populations and see what kind of responses would be. So these are the three components, right? Variation must occur. Members of the population vary in the traits they display. The variation that we see here in these beetles, right, is coloration. Is there anything else that's different among these individuals? Size, okay. So those are the two traits we could look at, coloration and size. We imagine that, and we'll be talking a lot about Mendelian, you'll be doing a dreaded Mendelian genetics lab, and we'll be looking at other kinds of traits in inheritance, but offspring tend to resemble their parents, right? so that these kinds of parents give rise to similar looking phenotypes and so on and so forth. So we assume there's some sort of genetic basis to the variation in the trait. And then what we call differential reproductive success. And what's the agent of selection in this particular example? It's a bird, a predator. This is a blue jay, right? So the idea here is that these brighter beetles, the idea here is the brighter beetles are bitter. Remember I was talking about those plants? These are, yeah, so brighter beetles are bitter and predators learn to avoid them. Again, bright coloration is, the predator associates that bright coloration with toxicity and a bad thing to eat. There are lots of studies where they do that and the bird gets them and they're naive and young and they eat one and then they throw up. Okay, and they learn very quickly, I better not eat that brightly colored one because it's associated with a nasty chemical compound that's a defensive compound against predation. So brighter beetles are bitter and predators learn to avoid them. Bright beetles are more likely to survive and more likely to reproduce than are the duller colored beetles. Under this kind of situation, the outcome's pretty simple. What happens, don't worry about this, we call this aposematic coloration. Anytime a bright coloration is associated with toxicity, it's usually a signal to or cue to something that wants to eat it, to avoid it, and it's a warning, what we call a warning coloration. This is very common in nature too. So here's the original population. What happens over time? Boom, boom. Evolution by natural selection. The proportion of different color variants in the beetle population changes over time. This is an example. What happens to the yellow beetles? They get eaten. They're lost. So this leads to the fixation of this particular kind of phenotype and the elimination of the other one. So you lose variation, and it results in a phenotype that has a higher fitness and is, I'm not going to say it. Okay. Now, was size acted on here, as far as you can tell, the way this is demonstrated? No. It's hard to say, but again, it didn't seem to matter, so you still have some smaller beetles and larger beetles. So what you would argue there is, did predators show any preference for different sized beetles? Maybe not. Probably not, at least in this example. So it doesn't really matter if you're red and toxic. So that's all natural selection is. Key components. Variation must be inheritable, and it has to lead to differential reproductive success, or what we call fitness, right? Phenotypes vary in the number of offspring that they pass on to future generations, and we'll be going through a lot of, that's what we call fitness. So before we get into the example of the example of natural selection and coat coloration in these beech mice, I want to talk a little bit about the fact that phenotypes that you see often aren't fixed, and the environment can influence the expression and the production of phenotypes, and this is extremely common. We'll be spending a lot more time on this later on when we get to what we call heritability and quantitative genetics, but phenotypic expression in general, the phenotype that's observable, that whatever we're looking at, in this case we'll be looking at the number of leaves and the mean leaf area of this particular plant. I think this is, I don't know what it is, purple leaf, some sort of, is it foxglove? Okay. How do you know that? Oh. Okay. Okay. Good. I should read the textbook ahead of time. Okay. I think it's in there. Yep. So phenotypic, well, our idea here is that phenotypic expression depends, we normally believe that phenotypic expression depends on both genes and environment. Some genotypes lead directly to a certain kind of phenotype, but oftentimes we see an interaction, what we call, between the gene and environment. Well, let's look at some examples of this. And the easiest way to do this is in organisms in which you can kind of clone or create genotypes, and that unique genotypes, that's real easy in plants through cuttings and all sorts of things. So what each line represents, right here, here's a red, blue, lighter blue, green. Each line represents a particular, it's a cloned individual of the same genotype. And so you're growing individuals of the same genotype across a range of light intensities, from fairly low light availability, intermediate, to extremely high light intensity. So the environment that we're looking at is the sort of light intensity availability of light. And what you see is, what do you notice here? What happens as light intensity goes up in terms of number of leaves? It increases. That's not surprising, right? Okay. There's more light availability. They have more light for photosynthetic resource, and so they can allocate a lot more to leaf, the number of leaves that they're producing. Okay. So clearly, the environment influences the expression, of the number of leaves. What happens to particular genotypes? What happens to them? So they all kind of go up, don't they? Okay. So one of the things you might want, what you'll notice here is that here's a genotype that kind of has the smallest response. Do you see that? Okay. Here's another one. It's hard to say, but a lot of these lines are crossing. Okay, and we're going to come back to this. But anytime you have this case where a genotype does well in one, produces a kind of value in one environment and a different value in another environment, but it varies among the genotypes, that's what we call a gene-environment interaction. So let me just give you an example of that. And we'll be coming back to a lot of these things. So here's environment 2. Here's number of leaves. Oh, you can't see it? Okay. There you go. Sorry. Let's imagine you just did 2 genotypes, right? Okay. Here's one genotype that produces number of leaves down there. Another one produces it way up there. If there was no effect of the environment on the genotype, and you grew them in that environment too, what do you think, where would the lines go? So what we're saying, if you had something like this, those 2 genotypes differ, right, in terms of the number of leaves that they produce. Does it depend on the environment? Not at all. It's a really fixed trait. The environment, the 2 genotypes differ in terms of their leaf production, but the environment has no influence on their expression. Okay? So that's what we call a fixed or canalized trait. The gene is completely influencing that phenotype. Huh? What's that? Yeah, don't worry about that. I throw those things out. That's it. Yeah, that's what, don't worry about it. Okay, let's come up with something else. Oh, jeez, I swear. Okay. How would you like those lines to go if you, what we call a gene by environment interaction, where a genotype and its relative position changes as a function of the environment? How would you like those lines to go? You want them to be parallel, even though they're not straight across? Do you want them going up like that, or you want them to cross? You want both? Oh, no, okay. Oh, jeez. I'll get it. That's what we call a gene by environment. Anytime these genotypes, their relative expression changes depending on the environment, okay? So what that's saying is genotype, this circle genotype produces few leaves in this environment, but overproduces these. This is what we call a gene by environment interaction. And again, we'll be talking we'll be talking about a lot of those things. So in this particular case, the environment influences the genotype quite a bit, right? And the relative position of the genotypes is strongly dependent on the environment. Okay. And you actually see, and look at the previous example, you do see some crossing of lines, don't you? So that would suggest that previous one is there is some gene-environment interaction in that other one. Let me just quickly, I get bogged down in these things, okay. So here's another thing that's important, and this deals with trade-offs. Look at this graph. What do you notice there? Yeah, what you see happens is, what happens in, let's just focus on the highlight environment. What we said is, man, you're producing a lot of leaves, but what's their relative size? Yeah. So what you're seeing here is it depends, but in many cases, some of these genotypes, the green one is they're producing a lot of leaves, but much smaller, okay? And this is an extremely common phenomenon in nature. And this is what's called a size-number trade-off, right? So that as you start producing a high number of things, often the size is kind of affected. And this is what we'll be talking a lot about what are called trade-offs. That you can produce a lot of leaves, but they tend to be, the mean leaf area is much smaller. And when you think about that, I'm not going to ask you this, but do you think that's a good thing in that highlight environment, as opposed to producing a small number of leaves of big size? That's a tough question. But in general, the idea here would be that this is actually the best response. To produce a lot of leaves, a smaller area, it's actually probably going to get you the ability to capture more light and produce more resources. Okay, I'll stop there. I just want to, we're gonna spend about, I might be able to get beyond this, but maybe not. This is one of my favorite examples of natural selection. This is done by Hopi Hoekstra, is a person who's been studying these in the beaches of Florida. So if you ever go to Destin or somewhere, look for them. They're all over there. She's been studying these for now about 20 years. She's at Harvard. She's probably one of the most famous evolutionary biologists in the country. And this is really, we're just highlighting some of her work. So again, she started with a simple observation. I think she was on vacation, right? In the beaches, and people knew this and already knew about it, and she went down there, and there are the phenotypes. And these are Polyostis, I think. It's a mouse. And what's different about those two on A and B? And the ones kind of below them? Yeah, one's darker, one's lighter. Real simple, okay? So you have a more melanic, darker colored mouse on the left and a much lighter, sort of yellowish pigment on the right. So we want to say, how did that happen? Yep, okay. So what it turns out, the simple observation is some of these populations are found in beach environments, sand dunes, beach environments, and the sand dunes associated with them. Some are more inland, often in old fields, sometimes in forested environments and those sorts of things. So the question is, how might natural selection favor different color variants, this sort of melanic and semi-low melanic mice in inland versus beach? So if you had to think about it, where do you think the lighter colored ones are? In which environment? Okay, straightforward, right? And the inland ones are the more melanic, darker ones, and that's the pattern, and we'll show you that. What do you think are can you think of some hypotheses involving natural selection that it would explain why those different color variants are favored in those two different kinds of environments?
The predator can see the darker ones on the beach, more on the beach, then the light will be more hidden.
Yep. So one is basically a camouflage hypothesis, and certain phenotypes have reduced predation by primarily aerial predators, hawks, owls, things like that. Okay? Those are the main predators on these things. Okay? So I'll tell you, that seems to be the answer. Can you think of an alternative, though? How? Yeah, and that probably goes along with, there might be mates, if a female is given a choice in an environment in the beach dunes between a darker male and a lighter male, selection would favor mate choice for the particular phenotypes that's better adapted. So those would probably go together. Okay, so that's good. I'm thinking of another, think about something else about the environment. Yep. Yep. So I would say a really good alternative hypothesis is, in those sandy beach environments, which one of those do you think is going to heat up faster? Yeah. So I think, and I'm not sure I haven't kept up with the literature. I'm not sure she's addressed that hypothesis, but I assume she has. Okay. But that might be an option. That might be an alternative explanation, or it could, those two hypotheses are not necessarily mutually exclusive either, are they? It's possible they work in concert. So the other idea might be these less melanic beach mice might not take on as much heat or overheat as quickly. So in general, there's better support for the original hypothesis. So how do we, I'm not doing that. Okay. That's what I do. I put iClicker questions in the middle of it. So, okay. So here's the distribution, just kind of showing you where this study was done. So in red are all of the sort of beach populations. And there are a whole series of them. They're along the Atlantic coast and along the Gulf Coast, and they tend to be kind of they're different beach mouse populations that are kind of isolated from one another. Their ranges don't necessarily overlap. And one of the kind of interesting things, I don't know how much has been done, but even within these beach populations, do they all look the same? No. You do see, you see this one's really light on Santa Rosa. In Perdido Key, it's a little darker. Over here, it's in Choctawhatchee, this is a little darker. Okay, so there's, even within what we call the beach mice, the less melanic ones, there's a fair amount of variability. And even within populations, you find, and I'm not going to go through all the details, but even within populations, you find some variability in the coloration. Pattern here, this is kind of the old field mouse, okay? These are the ones that are kind of in these gray areas all throughout this range that are separated a little bit, not so much here, but are, in these sort of old fields, perhaps more forested habitats, and that's the coloration that you see on those. So those are the very melanic ones. The pallid, this one's really light, and all these sorts of things. So, okay. So here what you see is, this is pretty remarkable divergence in these phenotypes, and I'm not going to go through the phylogenetic tree, but she actually did a phylogeny based on DNA sequence data, and it turns out all of these mouse beach populations arose. All of these populations here, and I think these two over here, all share a common ancestor. So remember how we showed that tree? All of those populations group in one, all come back to a single common ancestor, and it occurred about 3,000 years ago. So that divergence, that change has occurred very quickly. There are ways to date trees based on molecular data and what are called molecular clocks. And so they all originated 3,000 years ago, and that's when selection started to occur. So fairly recent evolution of these phenotypes. So what do we know about the genetic basis of these things? This is the even better part. Again, I'm not asking, you're not going to have to know all the details about melanocyte follicle stimulating hormone, right? I'm not too worried about that. But there's a gene it's called MC1R, is a major receptor that influences coloration in a lot of mammals and birds. It is extremely well characterized. We have the sequence data on all these genes in a whole range of organisms at the nucleotide level. We know what's happening in the coding. We know the mutations that are occurring. These have all been mapped onto trees. It's one of the best data sets we have. So what it is, it's a major receptor that influences coloration in many mammals and birds. You don't have to worry about too many details. Here's the receptor on the cell, and there's what's called alpha melanocyte stimulating hormone that has to bind to that receptor, or it can. So when that stimulating hormone binds the MC1R, it actually, when that happens, that stimulating hormone binds to the particular receptor of that MC1R, that results in what's called eumelanin. That's the typical dark coloration. So if you have that hormone binding to that receptor, you get darker mice. And this is true across all kinds of mammals and birds. A lot of this coloration is attributable to this particular gene. In the absence of this stimulating hormone binding, right, here you don't see that thing, what do you get? It goes in this pathway and produces pheomelanin, which basically results in a much yellower pigment, lighter pigment, and that sort of thing. So that's the basic pathway by which these phenotypes have diverged, and that's kind of the molecular mechanism. What's really interesting is there are actually two ways you can do that. Again, I'm not going to worry, you're not going to have to know that. But it turns out that what happens is some of these genes, this MC1R mutation, and they know what part of the coding region is mutated. It actually, these mutant 1CR receptors have a non-functional binding site. So there's been a mutation in some of these beach populations, and that thing can no longer bind to that receptor. So what do they produce? Light mice. There's actually then another gene, don't have to worry about the mutation in the agouti gene, it increases this expression, blah, blah, blah. This thing competes with the brown one, right? It actually binds, and guess what? It goes to that pathway. So they know the exact genetic mechanisms that result in these non-melanic phenotypes. And there are two, and the key point is not all these details, but there are two pathways that lead to the production of these less melanic types, these beach mice. And and it is a very simple series of single genetic mutations that result in that coloration change. Okay, now what do you want to know? So you got variation, you know that it's a heritable trait, it's due to two possible mutations, right? Are the what? I don't know. The ears all look the same to me. Oh, he looks kind of curly. I don't know. I don't think so. I have no idea. Well, what do you want to do now? You guys all think it's predator, right? There were a lot of stuff that was done earlier on that's in your book. Well, you think it's a predator, right? You would expect this predation rates to vary across these environments, right, depending on the phenotype. These aren't real mice. Those are silicone mice that she put out hundreds of them in the environment, and some of them were light coloration, some were darker coloration, melanic, non-melanic, okay? And she put them in the two different environments, and then she came back 10 to 20 days later, and why do you think she chose silicone? Because she could tell when hawks or owls grab a mouse, right? They grab with their talons. And I think most of them figured out as soon as they grabbed it or bit into it that this ain't real. And they dropped it, okay? So she looked at the proportion of attacks, right, by looking at the marks on these silicone models of different coloration in these different environments. And pretty simple. Do her data support the predation hypothesis? So here's an experiment that was done in the light background beach environment. Which phenotypes had a higher attack rate? Yeah, the darker ones, about 75% versus about maybe 25%, so a big difference. Did you get just the opposite pattern in the dark environment and the inland environment? A much higher attack rate on the light mice compared to the dark mice. Okay? So these are, I mean, to me, these seem like really simple things, right? But you have no idea how long this took to gather these series of data and this evidence. So basically what happens here is all of the key components suggest that natural selection via predation acting on a genetic mutation on this particular trait led to the pretty rapid evolution of differences between these populations. Okay? We might then ask ourselves at some point, are these different species and those sorts of things, and that's something we'll come to. But this really, I think, is one of the best examples of how selection can really rapidly influence that. I'm doing okay. Should I keep going? Do you get saturated about 60 minutes? Okay, let's go through this one just as an example. And when we think about adaptations, okay, what's an adaptation? Let's go back to the mouse example. What? Yeah, it's basically the idea that a particular trait or feature of an organism is somehow appropriate or allows higher reproduction or, capability to survive and reproduce in a particular kind of environment. And that's what we call an adaptation, okay? And there are all sorts of features on organisms. And the easiest way to look at them is organisms that live in extreme environments and organisms that don't have the capacity to really avoid extreme environments, okay? And an example are desert plants. One of the other greatest examples of people that are really looking at right now are deep sea thermal vents in oceans, right? That are full of sulfides, methanes, no light, high pressure, and it is remarkable, the series of adaptations, and remarkable the number of organisms that live there and the diversity there. But anyway, if we just look at, this is a saguaro cactus, Sonoran Desert, right? Ever been in Arizona, northern Mexico, Southern California, you'll see these sorts of things. So they have, and again, it's really easy to say, oh, these are all adaptations. So, and we'll come back to this, but obviously, what is the key thing that many desert plants face? What challenge do they primarily face? Yeah, lack of water and a very dehydrating environment, right? Especially as a plant. So you can imagine these have extremely high temperatures. I used to work in Chihuahua, Mexico, in a desert out in the middle of nowhere, and it was 120 degrees. And it's a challenging place. But it's a dry heat. I always love that expression. But at high noon in the middle of the desert, it's still not very pleasant. Okay, so anyway, so basically here are a series of what we might consider various kinds of adaptations that the saguaro cactus might have. Obviously it has flowers to enhance pollinators for reproduction. It has a shallow root system that collects water after infrequent rains, okay, rather than sort of a deep penetrating root system. Maybe that's a more efficient way to do that. They often have a waxy cuticle on here that prevents water loss, and they have all sorts of photosynthetic mechanisms that reduce water loss and all sorts of things. And then they have these sort of accordion-like structure that allows the plant to expand and contract with water availability and where they store water. They also apparently have sharp spines here, along here. Nobody crawls up them. Nobody wants to eat them. And apparently that is something that deters herbivores. Okay? So it's really easy to say that, right? Those are all a suite of adaptations that evolved by natural selection to deal with this sort of extremely dry environment. What I want to suggest to you is you shouldn't necessarily believe that. And what you'd really want to do is actually experimentally test that, okay? So let's look at this one example. So you can create any sort of story and say it's an adaptation, but sharp spines deter herbivores, things that are eating it. How would you test that? This is what ecologists do all the time, right? They're called removal or ablation experiments, right? You take a trait, you get rid of it, and then you look at is there a response. And there are all sorts of control things you have to do in terms of experiments, but that would be one thing you'd probably want to do to see is, you probably don't want to do it to the entire plant, but you might take, paired sides, and remove on this side, keep that, and look at what happens to fungal infection. Maybe it's not herbivores, right? I suspect it probably is, but it might be, maybe if you remove the spines, they do suffer from more sort of ingestion by an herbivore. Okay, so that's one way you can do it. Okay, I'm going to go through this, and what we're going to do is we're going to come back the next time, and I'm going to ask you to come with an answer. Okay, or we might do a little breakout session. This is a great example. Textbook got this wrong. Okay, I have no idea why they did, and they haven't corrected it, but okay. This is another about a 30-year study by David Resnick and a group of people in California in Trinidad and Tobago. Does anybody know where that is? If not, go there. I would suggest it. It's off the coast of South America, a couple islands, right? It's beautiful. Anyway, I'm just going to tell you a couple things. Guppies live there, right? That's the thing they're looking at. It's a predator-prey interaction. So in the Trinidad and Tobago is full of mountains, right? And streams running from high elevations down to the coast, emptying into the Caribbean or Atlantic, depending on where you are. So you have these streams and all over, 10 to 12 big major stream systems, river systems in each of these islands. And the pattern that we see here is replicated in all these streams. In the lower elevations, as you get closer to the ocean, in these lower streams, you find this cichlid predator that eats guppies. Okay, so in the low elevation environments, you find this thing. In the upper elevations, you find a predator. Guppies are all over, right? But this predator up here, Rivulus, is smaller, and it lives up there. Okay. So they got this kind of wrong. So these individuals up in these populations, they call them low predation sites. That's not necessarily true. This predator is there, and it does have, it's not as voracious, it's not as nasty as this cichlid, but it still eats things, don't get me wrong. So this thing up there, this predation site up in these upper marches, this predator eats prey. It turns out what it eats is, in these environments, this predator specializes on smaller juvenile guppies, and it leaves the adult guppies alone. Okay, so keep that in mind. You go down here, guess what this thing likes to eat in terms of the size and age of guppies? It tends to eat the adult guppies, the reproductively mature guppies, the larger guppies. That's its preference. In your book, it is right. Down here, these are much more voracious. The guppies actually school a lot more, form more schools, and that's what your book associates on, to kind of a defensive behavior. These things up here, these guppies don't do as much sort of schooling behavior to avoid predation. So there are big differences between these populations. Okay, so what happens to these guppies? So it's very different kinds of predation, right? This thing it's pretty voracious. It eats more adult guppies. These are good predators, but they tend to eat more juvenile, smaller guppies. Let me just tell you what happens. In these these sites where this thing's eating larger guppies, the females produce a lot and smaller offspring. So let's say that one produces 100 offspring on average, and they're 5 millimeters in size. Let's make something up, right? You go up here, guess what they do? They produce fewer offspring. So what did I say, 100, what did I say, 105? So maybe they produce 20, but they're much bigger. They're 10 millimeters in size. So again, this is what we call a trade-off, right? So what's happening here in the high predation sites down here, the number of offspring is high. What's their size, relatively speaking? Small. You go up here, what you have here is fewer, so this is 0, high number of offspring, small offspring, smaller offspring, larger offspring. You go up here in the low predation sites, what happens? They're producing fewer offspring, but much larger. Okay? So we're going to come back and try to explain that. What I'd like you to think about is why is that happening, and why is selection favoring that? The other thing I want you to think about, and I'll email it to you, what I want you to think about is how much should females invest in reproduction during a breeding season? Okay? So you're an adult female down in here, right? Should you invest in a lot in reproduction, or should you invest less in reproduction relative to this other population? So this is a very hard thing to think about, but think about this. What would selection favor? A female investing a lot of energy in reproduction in this population or less? So let me phrase these questions and I'll email it to you. And what we'll do is we'll come back at the beginning of class and I'll just ask, maybe you can just shout out some answers and we'll talk through how we think about those things. And this is what we call life history evolution, and it's another thing we'll talk about. So, okay, we'll stop there.